Hello, ladies and gentlemen. My name is Jeremy Webb. I'm the Editor-in-Chief of New Scientist. So welcome to New Scientist Live. Our speaker tonight is the well-known theoretical physicist and cosmologist, Professor Paul Davies, who is Director of the Center for Fundamental Concepts in Science at Arizona State University. Paul's career has taken him from Cambridge University to King's College, just down the road from here, to Newcastle University, and then to Adelaide, Macquarie Universities in Australia, before he moved to the US. Paul is not only an exceptional physicist, he's also, as you're going to find out this evening, a superb communicator of science. Now, I've known Paul for 30 years, I worked out yesterday. He probably won't remember it, but I first met him when I was an eager young producer at the BBC Radio's Science Unit in 1983. And I was introduced to this erudite professor of physics from Newcastle who didn't sound one bit like a Geordie. I still have a copy of one of Paul's books from that period on my shelf at home. It's called Superforce, and I thoroughly enjoyed it at the time. And since then, Paul has written many other best-selling books, including The Mind of God, About Time, How to Build a Time Machine, The Fifth Miracle, and The Goldilocks Enigma. He's also a recipient of the £1 Million Templeton Prize for his work as an author. His latest book, The Eerie Silence, is about the search for intelligent life on other planets. And this change of direction rather reflects a change in Paul's interests over the past 15 years or so, to include astrobiology, life beyond Earth. And Paul has been a major mover in revitalizing this field, I would say, over the last few years. And the change of direction highlights one of Paul's really critical skills. He has the ability to ask questions that cut to the heart of a matter, whatever the subject area. And he was asked, it's a rare skill, and one that has opened another door for him in just the last few years. He was asked by the US National Cancer Institute to propose some fresh questions to ask about cancer, which is why he's here today. I won't go into more detail because I'm sure Paul will tell us the story. So please give a warm welcome to our speaker tonight, Professor Paul Davies.
Well, thank you, Jeremy, for that kind introduction. Very warm welcome to you, ladies and gentlemen, especially warm welcome to the students in the audience. You are the future. We need all the scientists we can get, and especially in the subject I should be talking about this evening. It's great to be back in London, just 1 mile from where I was born, as it turns out. My life having taken me from the south of England to that totally alien country called Newcastle upon Tyne, and thereafter to Australia and the United States. Well, 42 years ago, US President Richard Nixon declared war on cancer. And in a landmark piece of legislation, he released the greatest ever research funding to tackle this terrible disease. It came at a time when progress against many infectious diseases have been spectacular, and he predicted that within a few years, cancer would be under control. I've said the biggest spending release. Currently, the National Cancer Institute in the US, these are US figures only, spends about $5 billion, that's billion, not million, $5 billion a year, that's taxpayer money. And incidentally, that's a little more than NASA spends on space exploration. Other organizations spend several billion of their own. And so in the time since Nixon declared war on cancer, and the 4 decades have elapsed, about $100 billion has been spent on cancer research in the United States. And there must be a comparable amount in other countries as well. So this is a staggering amount of money. And in addition to the cost of the research is the cost of the treatment. Now, that's a complex thing, particularly in the United States, but all I want to do is to draw your attention to the cost of drug treatments for cancer, which can be as much as $150,000 a year per person. I'm grateful to my colleague George Post for providing these numbers here. So this is a public health issue and a financial issue of spectacular proportions. Now, how are we getting on? 42 years later, all this money spent, how are we actually getting on in terms of what really matters, which is patient outcomes? With this graph here, it's a little out of date, but the story hasn't changed much, tells it all. If you look at the other major killers, say cardiovascular, then progress in that period of time has been truly spectacular. But cancer has hardly changed. So the death rates really haven't been impacted much by all of this spending. This is another graph. This is survival rates. Survival in the subject of cancer has a rather curious definition. It means if you're still alive 5 years after you were diagnosed. But of course, as screening programs improve, people get diagnosed earlier, and so it looks like there are more survivors, but it's a trick of the statistics. And you see, in any case, this isn't exactly spectacular progress. It would be wrong to say there's been no progress. Childhood leukemia has been pretty much cracked. So here and there are some conspicuous successes, but the overall picture for the major categories of cancer, breast cancer, colon cancer, lung cancer, and prostate cancer, the prospects of really, of somebody now who's diagnosed with metastatic cancer, the prospects are really not a lot different from what they were several decades ago, which is pretty shocking considering the amount of effort that has been put into this field. And the other chilling statistic that I got from my colleague David Agus is that the average increase in life expectancy for metastatic cancer, somebody diagnosed with metastatic disease, I'm going to explain that in a moment, with all of the miracle drugs, almost every day you open the newspaper, cancer breakthrough. We never seem to see those breakthroughs translated into patient outcomes. The average increase in life expectancy is 4.1 weeks. So cancer drugs prolong, mostly they don't work, most people, but when they do work, they prolong life for weeks or months rather than years. So this is a pretty dismal picture and a very expensive one. So we've got to do better. We need to come up with something new. Now, there's been about 1 million papers published in cancer research, and a couple of weeks ago I came across this on a website. Number of published cancer studies that can't be reproduced is shockingly high. In other words, wrong. The results are probably wrong. And you might wonder what is shockingly high. My ASU colleague, former deputy director of the National Cancer Institute, gave a lecture in April in which he said, by their count, 80% of cancer papers, 80% of these 1 million papers are actually wrong. So the field is in a mess, not for want of trying, not for want of money, but clearly we need to do better. And the same deputy director of the National Cancer Institute about 6 or 7 years ago called me out of the blue, and she said, you don't know me, but we're spending $5 billion a year of taxpayer money on cancer research, and we're getting nowhere. You physicists seem to be really good at solving complex problems. You've figured out what's happening inside atoms and at the edge of the universe. Can you help us take on the cancer challenge? And I said, well, I don't know anything about cancer. And she said, perfect. And thus began the creation of 12 centers for physical science and cancer and oncology, or cancer biology. And here they are. I'm running the one at Arizona State University, ASU, there. Scattered all across the country, 12 centers in total, annual budget about $35 million set up on a 5-year program. I'm not going to go too much into these organizational things, except to say that I'd never before dealt with US government agencies, and I'm not sure I ever want to again. Our particular center, which we call the Center for the Convergence of Physical Science and Cancer Biology, I'm the principal investigator, and so I'm running this particular show. We have about 30 people in total. we are twinned with the Mayo Clinic in Scottsdale and the Fred Hutchinson Cancer Research Center in Seattle. We call that the Hutch. All up, we've got on our team 6 clinical oncologists or cancer biologists, 5 physicists, a physical chemist, an astrobiologist, 3 engineers or nanotechnologists, 1 person, that's my wife as it happens, dealing with education and outreach, and about 15 students. And that's our website. I'm going to keep putting it up because I'd like you to go there. What I'm going to talk about tonight is my personal take on the subject of cancer, because I didn't just want to run this thing, I wanted to have some intellectual input. And I've been doing a lot of reading, a lot of talking, and a lot of thinking. And the thinking part is the most important in the 4 years since we've been up and running. And I really think that I have something new to say about the subject of cancer, although I found in this subject, biology is not like physics. Things are rarely black and white. So mostly, if I make a statement about cancer biology, an oncologist somewhere will say, oh, well, we knew that. And so the point is not what does the collective community of cancer biologists know, but are they connecting the dots in the right way? And in biology, there's much more narrative than there is firm principle. And so people tell stories. They have ideas about this gene or that pathway, this type of cancer, that type of cancer, and they're narratives. And there's a whole lot of narratives competing. Very rarely do you have any proper theory or anything with predictive power. Now, what I have to say this evening has been done in collaboration, most especially with Charlie Lineweaver there. He's at the Australian National University. My postdoc, Sarah Walker, she's a NASA astrobiology postdoc. My other postdoc, Luis Cisneros, and my wife, Pauline, who looks after the education and outreach, but is also unfailingly critical of every claim I make and reads everything I write, and she won't let me get away with total nonsense. So I hope this evening I won't be speaking total nonsense, or she hasn't done her job properly. I'd also like to thank the NASA Astrobiology Institute. We're collaborating with them, and of course the National Cancer Institute, who are paying the bills. Okay, well, let me get into what I really want to talk about. Oh, one last thing. Arizona State University is a bit of a sort of cancer cluster there. We've got some superstars. We have Lee Hartwell, who's a Nobel Prize winner, cancer biologist. We have George Post, former head of research at GlaxoSmithKline. Anna Barker, I've mentioned, the former deputy director of the National Cancer Institute. And we just acquired Ray Dubois, you might think, but over there it's Du Bois. And he was running the MD Anderson Cancer Center in Texas. So a lot of people, these are not formally part of our Physical Science Oncology Center, but they're close colleagues and keep me in line. Now, the point I want to get across right at the outset is, in spite of all these billions of dollars and all these years of research and huge amount of effort, there's no actual theory of cancer. People are so fixated on just trying to cure the disease that they don't stop to think, what is cancer? Why is it there in the first place? And how does it fit into the great sweep of evolutionary history? And so, being a scientist interested in fundamental principles and deep ideas, those were immediately the questions I asked. Well, what is this thing called cancer? The take-home message, in case you want to get up and walk out, is that we're not going to cure cancer. Never use the C word, the cure word, because I just think it's distracting. And we don't need to cure it for reasons I shall explain. I think cancer is a bit like aging. If I say to you, well, I travel all over the world, and everywhere I go, there's aging. You see it everywhere. People are dying of it in their millions in every country. We've got to do something about it. We've got to find a pill to make it go away. Well, you'd think that would be complete nonsense. However, if I say people are living longer, so senescence, aging is a major part of life, we must mitigate its effects. There are all sorts of ways of mitigating the effects of aging. In the same way, we can mitigate the effects of cancer. We don't have to just say, well, there's nothing we can do about it. There is something we can do about it, but it's not so much a disease to be cured as a condition to be managed. Sometimes physicians refer to that as a chronic disease. I don't even like the word disease because it's a condition. But we don't have to argue over that point. So what then is cancer? So for some of you here who might not know, only in the vaguest terms know what it is, it's very simple. And people often say cancer is a complex disease. It's not. It actually is a very simple disease to describe. It's a weird disease, but it's very simple to describe. The basic story is cells start to proliferate uncontrollably in one particular organ, and then they spread around the body and colonize other organs. And 90% of deaths attributed to cancer are from this spreading around process called metastasis. And you might think that there would be a pretty good understanding of why cells actually do this, why they start to proliferate uncontrollably and spread and go and colonize other places. There isn't. I'm hoping to give you one. Metastasis requires many steps. It's not just a matter of, well, they're here 1 minute and then they spread around. Each organ of your body has cells, somatic cells, which are very specific, terminally differentiated, to use the jargon, which means that they are particular types. A liver cell is not the same as a kidney cell, is not the same as a brain cell, not the same as a muscle cell. They're all different types. But with cancer, the cells can start out as one type and then they can go live in an organ of a different type, something that's more or less impossible. If you put a kidney cell in the lung, it doesn't do very well. Cancer does just fine. So this is what happens. You get a primary tumor in some organ. It maybe sits there for a long time, but eventually it gets too big. It hasn't got enough oxygen, and so it generates its own blood supply. That's called angiogenesis. And then that doesn't matter so much, but then the really shocking thing happens is that the cells detach themselves, some of them do, and they go on a journey. They, instead of being anchored, they suddenly become mobile, or motile is the correct term, mesenchymal cells is what biologists call it. It took me ages to pronounce that word. So these are, mostly cancers arise in epithelial cells, so these are like inner linings of ducts and tubes and things inside you, and they become mesenchymal, and they move around the body, and then they do this by either through traveling through the bloodstream or the lymphatic system. So they have to get into it. That's called intravasation. It's another one, took me a long time to learn how to pronounce. Intravasation, get into the bloodstream, they get swept along in this raging torrent, and then they end up extravasating. That is, that they get out of the bloodstream, sometimes with a lot of effort, and there's a lot of physics, interesting physics involved as they batter and squeeze their way out into other tissues, and then they can form secondary cancers, and that's the really dangerous step. On the way, or when they get there, they recruit healthy cells from the tissue microenvironment that they've made a home. There's evidence they even prepare a niche ahead of time. That is, that the primary tumor sends out spies or scouts or fertilizer, many metaphors, to prepare the ground for the nest that the colonists are going to make. So when they arrive, they've got what they need. So there's a lot of signaling. There's a whole network of signaling between the primary tumor and the metastatic tumors. If we had the chemical equivalent of the electromagnetic pulse that you may remember from the dark days of the Cold War, this was a nuclear explosion would knock out all your electronics in one hit. We had the electromagnetic pulse to knock out that command and control system, then maybe you'd knock out the whole thing in one go. That's not what I'm advocating, by the way. It's just occurred to me in passing. Okay, so that's the basic story of cancer. Cancer has a number of hallmarks. There's a famous paper by Hanahan and Weinberg I can recommend, and it got updated for the anniversary. The point is that cancer can be recognized by the changes that have taken place in the cell, and you probably can't read that there, so what I've done is to prepare another slide. These are the clever tricks that cancer does to survive, not only survive, but thrive in the host organism. First thing is that they switch off apoptosis, that's programmed cell death. Most cells in your body can replicate only a handful of times and then they have to commit suicide. That's programmed into them. Cancer cells switch that off, that's why they proliferate. There are genes that are meant to stop this happening, tumor suppressor genes, they switch those off. They convert their metabolism to fermentation or glycolysis. instead of using the normal oxidation phosphorylation that takes place in the mitochondria, healthy cells. I'm going to come back to that. They become squishier, softer. This may help them squeeze through little gaps, but they definitely become softer. One of our experimental research programs is to measure the Young's modulus, the elasticity of cancer cells as a function of cancer progression. They secrete, how do they burrow into bloodstreams? They secrete chemicals that dissolve away the membranes. So they can get through the basement membranes of organs and get through membranes into blood vessels. So they've got all that equipment. They go from being static to motile. I think I've told you that. They grow in distant organs. They recruit non-cancer cells. I've said all these things. They can organize their own blood supply. They can trick the immune system. The immune system's quite well tuned to recognize cancer as being undesirable and can knock it out. But cancer can evade this. And then, of course, when it gets to the stage of drug treatment, then cancer is notoriously resistant to drugs, which is why they don't work very well, because they just evolve resistance. So that's the list. Now, the point is that cancer doesn't invent any of these tricks. The body already does all of these things. For example, extravasation, cells getting out of the bloodstream into the organ. That happens as part of normal wound healing or inflammatory response by the immune system. So what cancer does, it co-opts what it needs to enact this agenda. And the point is that it's not just one of these properties, it's the co-presence of all of them all together that is so significant. And all this is done with what I call monster cells, because when you look at cancer cells, one of our projects that I'm in charge of is to get 3D images of cancer cells with a cell CT scan. I won't go into the details. They get all bent out of shape. So the morphology of the cells changes and the morphology of the nuclei changes. And within the nuclei are the chromosomes made of chromatin. This chromatin is distributed in a characteristic way in healthy cells. It gets distributed in a very different way in cancer cells. So the whole architecture of that chromatin, which relates to the ability of the cell to read out the genetic information, is transformed. So these cells look like they're a mess. They look like monster cells. And sometimes when you look at the chromosomes, they are a mess. There's a phenomenon called aneuploidy, which means the wrong number of chromosomes. Sometimes you get whole chunks of chromosomes transposed, and sometimes they're duplicated. Sometimes you get entire duplication of the whole set. That's called tetraploidy. All of these things are seen in cancer. So at first sight, you think these are monster cells. They shouldn't survive at all. How on earth do they keep running, let alone be the invincible stormtroopers that it looks like they are? So when I saw all these facts and asked, why is cancer such a formidable adversary? What is the secret of its success? It looks too efficient and deterministic to be just an accident or a series of accidents. It looks like it's a pre-programmed response to some sort of stress. It looks like it's built into the system. Another way of putting it is that cells come preloaded with a cancer subroutine that can be triggered by a variety of insults. Because when you see something working really well like this, when it's deterministic and not random in its behavior, it looks like there's something going on, and we need to figure out what that something is. So I asked the question, why do all cells in the body, pretty much all of them, come preloaded with this, well, all of them, they all have the same DNA, come preloaded with a cancer subroutine? Why has life got a cancer subroutine in it that can potentially destroy the host? How do we answer that question? Well, I'm a physicist, I'm not a biologist, but I know that in biology there's a famous phrase due to Theodore Dobzhansky that nothing in biology makes sense except in the light of evolution. And so we look to the theory of evolution to explain these facts. Now, because cancer touches every family on the planet, we're fixated on it as a human disease that we hope can be cured. But actually, cancer is extremely widespread in the animal kingdom. You see it in fish, and in all mammals, and in reptiles, and birds, and even a form of cancer in plants. So it pervades the biosphere. So this looks like it's not life gone wrong in some way, but a very deep-rooted, widespread, and therefore very ancient property of multicellular life. So to understand cancer, we need to understand, place it within this grand story of evolution of multicellular life. So the first conclusion then is cancer is not confined to humans, it's widespread in biology, so it must have deep and ancient evolutionary roots. Let me tell you something about these evolutionary roots. Cancer really is something that doesn't make a lot of sense except in the context of multicellularity. Unicellular life, single cells, which prevailed on Earth right up until about 1.5 billion years ago, life was just single cells. They've got a simple agenda: replicate, replicate, replicate. So that's what bacteria do, for example. Not a problem. Multicellular life is a much more complex proposition because in multicellular life, there's a contract that's struck between most cells, called somatic cells, the ones that you're looking at now, and the germ cells. Unicellular life is, in a sense, immortal. In multicellular life, immortality has been outsourced to these specialized germ cells. So it's retained in the germ line. When I say germ cells, some people say you mean diseases, germs. The germ line I'm using in the biological sense to mean the sex cells, the heritable information. And so multicellular life means that this contract has to be signed or struck so that the vast majority of cells in the community that makes up the multicellular organism accept mortality in return for their genes to be propagated through the germline. That's the deal. And there's obviously got to be some advantage in that. And cancer is clearly a breakdown in this ancient contract. And ancient because multicellularity goes back, as we'll see in a moment, well over 1 billion years. It's a breakdown in that contract because cancer cells are in effect saying, I don't care what you, the organism, is telling me what to do. I'm not going to apoptose today. I'm going to replicate. And then they embark on their own agenda. So it's a breakdown of that contract. There's nothing very controversial about that. I think most cancer biologists would agree. Multicellularity is a bit of a mystery because how did it happen? What is the selective advantage in biology, in the evolutionary sense, of becoming multicellular? Doesn't seem to be a lot in it, but there must be. The logic of multicellularity must be deep because it's happened independently many times. I don't know, this is a question, I don't know the answer. I wonder whether cancer is in fact an indispensable corollary of the pathway to multicellularity. In other words, the logic whereby multicellularity arises is also the logic that gives rise to cancer. I think cancer might be a hangover from the deal that got struck. Come back to that. You might say, well, we don't see cancer in these other groups like fungi, do we? Well, don't we? Has anybody actually looked for cancer analogues in fungi? I bet they haven't. And so I suspect there are cancer analogues in all these different families of multicellular organisms. Don't know, it's a conjecture. So this is a sort of homemade tree of life that my colleague Charlie Lineweaver lent me, and it's really just to make the point that I don't want to go through the whole history of life here. Leaker, that's the last eukaryotic common ancestor. Eukaryotic cells are the big complex ones that we're made of. So basically plants and fungi and jellyfish and so on split away in this period here. Earth is about 4.5 billion years old. There's been life on Earth for nearly 4 billion years. But most of the action I'm talking about starts between 2 and 1 billion years ago. And by about 600 million years ago, the multicellular life had become sufficiently rich and diverse that Stephen Jay Gould was able to wax lyrical about the Cambrian explosion. Turns out there was an earlier one called the Ediacaran explosion. Doesn't really matter. The point is that the differentiation of cells into many special types and many organ types and the large rise in complexity was pretty much firmly established by about 600 million years ago. So the story of cancer, I think, lies somewhere between 1.6 and 0.6 billion years ago, and that's where we have to trace its evolutionary roots. Now, I got onto this line of reasoning by reading a news item a few years ago about a tumor suppressor gene being discovered in a sponge. I never thought of a sponge as doing much, but it is a sort of animal, very simple one. I think if you went in a time machine and went back 1 billion years, you'd see a lot of sponge-like entities, a lot of tumor-like entities, rather similar in some ways. But the truth is that already, because of this deal that was struck, already sponges need to manage apoptosis, cell proliferation and tumor suppression. Here's the paper, and the authors, it's published in Nature, the authors say that the hallmarks of metazoan multicellularity are therefore intimately related to those of cancer. And that sort of leapt out of me. And so I've been developing these ideas ever since. Now you might think, well, if cancer is such bad news, why hasn't evolution got rid of it? Why do we still have to suffer from this? If it kills people, surely there's some selective advantage in getting rid of cancer. And the answer has got to be because it serves a useful purpose. Now, there can be many useful purposes for the cancer subroutine, but the one I'm going to dwell on for the next few minutes is a very useful one indeed, and it's this. It's embryogenesis. The reason cancer hasn't been eliminated is because the genes that are active in cancer are, in many cases, also the genes that are active during the development of the embryo. Now, you might think that this fact will be known to all cancer biologists. It will be the number one significant fact in research to try to do something about cancer. And although cancer biologists will tell you, yes, we've known this all along, the significance is really very much underrated. For example, here's a paper I dug out the other day after a Google search published in 1987 by Eileen Adamson, Oncogenes in Development, a very comprehensive list of those developmental genes that are reawakened in cancer. And here's another paper by Marilyn Monk, who's right here in London at University College, Human Embryonic Genes Re-expressed in Cancer Cells. You'd think this information would be screaming at us. Well, you won't find a great deal of work being done in this field. Developmental biologists don't talk to cancer biologists. They work in different departments, have different funding agencies, and different interests. But there's a little bit of work that continues. Isaac Kohana at MIT has been working on genes that are upregulated, I'm going to explain this in a moment, upregulated in cancer that are active during embryogenesis. And this is the quote: we identify a set of genes that are upregulated in most cancers, and we show this signature is active in early embryogenesis. And the more malignant the cancer, the earlier the stage of embryogenesis are those genes expressed. So cancer staging begins to make an appearance. It's the work of John Condelis. Don't expect to read all these technical details. I just want to mention him, the Albert Einstein Hospital in New York. And he too finds that the metastatic cells, the invasive migratory ones, have gene expression profiles which are reminiscent of, in fact, the same as cells in embryonic and tissue development. It's a really important fact. You can even grow embryos from cancer cell nuclei. These were mouse embryos, I think. Yep, grown from cancer cell, brain cancer cell nuclei. Now, this is where the biologist is going to walk out, because this is not politically correct. Ernst Haeckel in the 19th century coined this phrase, ontogeny recapitulates phylogeny. What does that mean? So ontogeny is the development of the embryo, the stages it goes through. And what he saw was, you look at a human embryo, early on it looks a bit like a fish, then it looks a bit like a monkey, and then later on it looks like a human. So that's not the right way to think about it. The right way to think about it is that across the animal kingdom, there are many resemblances between the embryos. So the various stages of embryo development correlate with each other. And that's not really surprising. because if you pull a brick out of the foundation of a house, it might fall down. But if you pull a brick out of the top of the chimney stack, that's okay. It's much easier. So biology always builds on what goes before. So if you want to tinker with something, you tinker with the recent stuff. You don't tinker with the basics. And because we share with these organisms the same basic body plan, it's no surprising that when that body plan is instantiated during the development of the embryo, it follows similar stages. So this isn't really surprising, but some people don't like this concept of ontogeny recapitulating phylogeny. Now, what this means is our genes retain, as it were, a record of the past, of the evolutionary past, because nature builds on what's gone before. And this can sometimes be spectacularly demonstrated in what are called atavisms. So an atavism is a mistake, but it's a tendency to revert to an ancestral developmental pathway and to lead to organs that our ancestors may have had, like tails and fins, a long time ago, but we don't generally have today. And so here's a dolphin with 4 fins, reflecting its 4-limbed ancestry. Here's some more striking ones. Boy with a tail, supernumerary nipples, so not as uncommon as you might think. Webbed feet, our ancestors were aquatic. And a whale there with arms and legs. So what happens is it's not that we've still got genes for fins or webbed feet. It's just that that all the genes we have all have a history, and these genes all go back. I'm told off for talking about ancient genes. You mustn't do that, because all genes, in a sense, have an ancient ancestry. It's just some genes are conserved and don't change very much and are widespread across the tree of life, and some change a lot. So the existence of atavisms just shows that we are retaining capabilities of ancestral phenotypes. So the phenotype is the expression of the genes in the physical organism. So the hypothesis that Lineweaver and I have developed then is that in cancer, as cells become deregulated, clearly this is what happens in cancer, that the body loses control over them, otherwise it wouldn't happen, become deregulated, we think they default back to ancestral genetic pathways. As it were, they appropriate genes which are these ancestral genes, conserved, widespread genes, and it's an ancestral phenotype. And these, by what I've just told you, these ancestral genes are the genes which are expressed most obviously in early stage embryogenesis. So this is in a sense life gone wrong, but what is happening is not random, not cells running amok, it's cells following an ancient program. So it's a little bit like this. You've probably seen this on your computer screen a few times. Safe mode. It happened to me last week when I dropped my computer. So a hardware insult or a software insult, a glitch, will result in the computer starting up in safe mode. So I'm thinking that cancer is a little bit like that, a cassette or a package of genes that are a bit like the safe mode. And if something goes wrong, well, let's reboot in safe mode. So the threads I'm trying to draw here are between cancer biology, developmental biology, and evolutionary biology, and I put in astrobiology there. What's this astrobiology? Are scientists who study life wherever it is. Doesn't have to be on Earth. Could be Mars or Europa or extrasolar planets. I'll be talking about that tomorrow at the Cheltenham Literary Festival. We haven't found any beyond Earth yet, but we can think about it, and that's good, because astrobiologists think about life in a very deep way, in a way that most biologists don't. And so they are always wrestling with the question, well, what is life? And how would you know it if you saw it? And what is the logic of life? And what is it trying to do? Very useful to have those people around in a subject like this. Now, to further develop our theory, we make a number of predictions. Predictions are unusual in biology. We're actually making a number of very specific predictions that can be tested. Because what we think is that most interest among oncologists, and indeed the general public, is have you got cancer or not? In other words, it's the initiation of the cancer. They're less concerned with studying the staging, the progression of the disease. But I think this is the crucial part. as cancer progresses, in effect, is running the arrow of evolution backwards in time at high speed. So here we have the, over 2 billion years, phylogenetic evolution. Here we have ontogeny, recapitulating in a few months the course of that evolution, roughly speaking. And here we have carcinogenesis, cancer progression, starting with benign warts and ending with death, running in this direction, backwards at high speed. I said that this leads to a number of testable predictions. I'll just mention a couple of things, but there are many. One of the things that biologists are very good at now is working out phylogenies for all sorts of properties of cells. Now, if you're a multicellular organism, it doesn't make any sense unless you stick with the crowd. And that is literally true in the case of you and I and sponges and other organisms. The cells stick to each other, and they do that because they're like Velcro on their surface. That Velcro, these are molecules called cadherins, and there's a huge number of cadherins. So bacteria have cadherins, we have cadherins. The tree of the cadherins has been worked out. There's 11 branch points between bacteria and us. All these different types of these cadherins. So we think that as cancer progresses, it defaults back from the cadherins used by the healthy cells. I think they're N, I never remember which way around, whether it's N or E, either N or E for healthy cells. We know, it's already been established, that cancer expresses the E cadherins, or the other way around, and we think that that can be a systematic relationship and could be correlated with this phylogeny here. So that's one definite prediction, the cadherin expression. You can get that from the gene expression profiles of cancer as a function of stage. The big problem is getting that clinical data. If you tell someone that they've been diagnosed with cancer, you don't say, well, we'd like to leave it alone and see how it progresses, and every couple of months we'd like to take a biopsy and get a gene expression profile, and that would be really useful for our scientists. So it's not the sort of data you can get. You might get it with leukemia, but for solid tumors, this is not really a very easy thing to come by. You can do it for mice. A lot of dead mice in this game, I can tell you. Anyway, so that's one particular property of cancer, the change in the cadherin expression. There's others having to do with pumps, for example, and we'd like to look at the phylogeny of the pump. So in all cases, all those traits, remember the hallmarks of cancer, all of those things mean that something is being expressed differently from healthy cells. And so we'd like to trace the phylogeny of that across the animal kingdom, all the way back, to see if it does correlate inversely. There's another very interesting story. This is Otto Warburg, often called Warburg, who, interesting character, Nobel Prize winner, he was Hitler's oncologist. So people don't like to talk about him. Yet he found out something very profound about cancer that remains true to this day, often called the Warburg effect. And it concerns the way cancer deals with oxygen. So I mentioned that normal cells use mitochondria to make ATP, the stuff of the energy that powers life. They do that with a process called oxidation phosphorylation. It uses oxygen, and it gives you ATP. Cancer cells have a different mode of making energy. It's called fermentation or glycolysis. They can cut out the mitochondria, so it's a big change. And this glycolysis isn't so efficient, but it's much faster. So if all you want is biomass, it's actually quite a good way to go, but it's a very inefficient way. And so they're greedy, and they run on sugar. So cancer loves sugar and hates oxygen. The byproduct of this fermentation is lactic acid, which lowers the pH, the acidity of the region around the cancer. So cancer loves sugar, hates oxygen, and likes acid. I've wondered for a while whether this predilection for low or hypoxic environments reflects the low oxygen conditions on Earth at the time that we're hypothesizing that cancer can be traced back to. origin of multicellularity. That occurred in the period before the first and second great oxygenation events. So you probably know that Earth came without any free oxygen in its atmosphere, and for a very long time that was the way things remained. Eventually photosynthetic bacteria built up oxygen in the atmosphere, but that oxygen all got sucked up, rusting everything, until that was saturated, and then eventually the oxygen did build up. And it was only a few 100 million years ago that this second great oxygenation event occurred. So early multicellular life wasn't dealing with large amounts of oxygen. And if it's true that cancer is a sort of reversion or an atavism or a throwback to that early time, then maybe its ability to deal so well with hypoxic and even anoxic, no oxygen, environments reflects that. It's a conjecture. I want to come on to why people get cancer in the first place. And one thing I'd like to dispel, because many people have a dread of cancer, I think everybody does, is this idea that you've either got cancer or you haven't. And if somebody's told you got cancer, it seems like a calamity. The truth is there's no dividing line between what life is doing with and without cancer. I mean, it's just a myth. It's not like you've caught a disease. It's a gradual transformation of cells. So some cancers, cancer of the esophagus in particular, where you can follow the transformation all the way through the stages from, healthy to a little bit unhealthy, a bit more unhealthy. It's a condition called Barrett's esophagus. We work with those patients and their biopsy samples. And there's no dividing line. And a very large number of cancer cells never cause a problem. Most tumors never cause a problem. Most primary tumors, even if they're big, only rarely cause a problem. It's when the cancer spreads around that it becomes problematic, for reasons that nobody really understands. But even when the cancer cells spread around, the vast majority of them never cause a problem. So it's better not to think, this is a calamity, I've got cancer. Pretty much everybody's got cancer. Certainly everybody my age is going to have lots of what you would call cancer cells in them. It doesn't mean they're going to become clinically relevant. And that's the big problem about testing and screening programs. You can identify a small tumor, typically a breast tumor identified from a mammogram. We know that most of those will never cause a clinical problem. They will never progress. But you don't know which ones. That's the difficulty. That's why we have to understand cancer staging and cancer progression with a theoretical basis, not just say, well, this is what we find, that's what we find, this is what we're told. We have to understand what's going on in cancer progression. I think it's the arrow of evolutionary time running backwards, and I think that gives us a handle for controlling it, but let me come on to that. Now, you'll often read that cancer is a result of genomic or genetic changes. What does that mean? There are two completely different types of changes. They often get conflated. The first of these is mutation. So all of your genes have a particular sequence of A's, G's, C's, and T's in the four-letter alphabet of life as we know it. Changes can occur in that sequence. For example, hit by a cosmic ray or damaged by some oxidating reactive oxygen species from some chemical or something. These sorts of things happening all the time. And life deals with them very well. It goes through and edits out the mistakes and repairs the damage. Sometimes it's overwhelmed. So that's one way in which genes can change. The other way is not all genes are on all the time. In fact, mostly they're off. Genes, every cell in your body has the same genes, but they're all different cells. So different genes are expressed. And genes are silenced by a process called methylation. I don't need to go into the detail, but they have the methyl group attached to them, and this disables them by a mechanism that we discovered in our center just last year. Discovered why methylation disables the readout machinery. So most genes are methylated. Most obvious is the X chromosome in women. Women have 2 X chromosomes, men have 1. 1 X chromosome in women is completely silenced for their lives, otherwise they're going to express twice the protein complement of those genes, which would be bad. So The term genetics is given to the first type. The term epigenetics, which is now increasingly important, is the trying to understand all that is beyond the genetics, including the gene switching. And furthermore, they switch each other on and off. They form gene regulatory networks, like the internet. So a gene switched on over here can switch off another one over there, and so on. And there's feedback loops, and it's very complicated topology of these things. And so nothing in biology is simple. It's not a simple cause and effect. And a lot of biologists seem to forget that. They seem to think that you can trace something back to a cause. That mutation caused cancer. Don't think it's true. So we've got two alternative theories of cancer. The standard one, this is one you read about in all the newspapers and the textbooks, is the error theory of cancer, or the bottom-up theory. It's a reductionist theory in the traditional philosophical sense, because it attributes the origin of cancer to genetic accidents, that is, mutations in certain genes, maybe an accumulation over a lifetime, 6 or 7 or something, and this somehow unleashes the cancer, and as the cancer progresses, more and more mutations appear. So if you look at full-blown metastatic cancer, it's an absolute nightmare. There are millions of cells, all completely different. Their genes are all over the place. Everything is a complete and utter mess. So that's the theory. But there's another one, which is a minority theory. It's the one that I'm more sympathetic to. But again, nothing in biology is black and white, so there's going to be a bit of both. But this is the stress response theory. This is a top-down theory. What this says is that if a cell is in a bad environment, could be bad for all sorts of reasons, that switches on or upregulates or triggers the cancer subroutine, and that the mutations that you see in cancer are almost all collateral damage, byproduct of the cancer, not the cause of the cancer, byproduct of the cancer. So it's inverting it. So, genomic change is cause or consequence. Let me just repeat this. The standard explanation is accumulating mutations by some mystery permit cells to acquire all at once all of those wonderful, useful hallmarks, those survival traits that I've been talking about. Isn't that lucky? In a neoplasm, which is a population of new cells, obviously there's some variation and some selection. So of course there'll be some Darwinism going on in the body over a period of months or years, however long the patient survives. Of course there'll be some variation. But I think this is just the fine print, an embellishment on the fundamental story. The fundamental story is that cancer is not running on defective genes. Cancer is running on perfectly good genes, an ancient cassette of perfectly functioning genes that are simply inappropriately upregulated. They're switched on when they should be switched off. Fine in the embryo, maybe in wound healing, but otherwise you want to keep them silent. So our theory with Lineweaver is that a range of insults, and this could be mutations, a mutation, for example, in a tumor suppressor gene, a random mutation that disabled the tumor suppressor gene, might switch on this cancer subroutine. So mutations can be a trigger, but a trigger is not the same as an origin. It's a trigger. And the genomic instability is the collateral damage. So the metaphor I like is the genie in the bottle. So this is a healthy cell. There's a cancer subroutine inside every one of your cells. That's the genie lurking in the bottle. The gene is not defective. The genie operates perfectly well. The genie has his own, I think genies are only ever male, aren't they? Genie has his own agenda, but so long as the bottle confines the genie, everything's fine. Now, that bottle can be broken by a very wide range of accidents. There are many ways of getting cancer, but once cancer starts, it follows a very predictable and deterministic trajectory. So, the way I see it is that concentrating on the mutations is concentrating on what broke the bottle, not what drives the genie. And so if we can understand that genie, what is driving the genie, the natural history of the genie, going back to the dawn of multicellularity, then I think we can learn to control the genie or get the genie back in the bottle. Now, Charlie prefers another metaphor. This is a slide of his, the palimpsest. So a palimpsest is where you have one message over the top of another, or one painting over the top of another. This is an example. Renaissance artists or medieval artists at least, were very fond of doing this. And each of them is coherent in its own way. So imagine going up to a perfectly wonderful painting and throwing turpentine on it at random. That's like the random mutations. And you might think that you create a random pattern. And the standard explanation is cancer is that random pattern. That's like looking for messages in tea leaves. It's not where the answer is. The answer lies in the pattern behind it that is exposed by accident. The pattern that is behind it is perfectly coherent, undamaged, and will run. It'll run a cancer subroutine. It's not supposed to do it in the adult form. So it's running a perfectly good program at the wrong time as a result of being randomly triggered. And because everyone's fixated on the trigger, we've got to find out what causes cancer. Is it this mutation? Is it that mutation? We look at all these different cancers. We see common patterns and so on. You're really focusing on the wrong thing. Of course, we would like to prevent those triggers from happening. But once they happen, what they unleash is very deterministic. So what do we do then? How do we manage cancer, if I'm even remotely right? Well, there's some obvious things. You should prevent it. So you know you mustn't smoke or lie around in the sun or overeat. Avoid inflammation. Take aspirin is a pretty good way. Taking a baby aspirin a day for people of my age is a pretty good anti-cancer drug. Or possibly vaccinate. One of my colleagues at ASU is trying to develop a general-purpose cancer vaccine. We can vaccinate against some infectious diseases, viral diseases that cause cancer, go on to trigger cancer. So basically anything that causes inflammation is a cancer risk. If you can vaccinate against that, classic example being hepatitis. We can now vaccinate against hepatitis. Huge number of liver cancers have been removed. We can treat Helicobacter pylori, with which lives in your gut, cause inflammation, and gives rise to stomach cancer. We can vaccinate against that now. So that's all fine. So obviously you prevent it, but once you've got it, what is the strategy? Well, I'm not a fan of beam weapons or toxins, which is the present, or surgery, which is the present method. I think we have to find a better way, but what is that? So this is where physics comes to the rescue. The point is that cancer cells are not invincible. Cancer cells are actually vulnerable. They suffer. What you need is something that will make them suffer more than healthy cells. And if I'm right, the cancer cells are a sort of atavism, then I've already said it. They love sugar and hate oxygen. So we have to give them lots of oxygen and deprive them of sugar and restore the pH. You can give lots of oxygen with something called hyperbaric oxygen therapy. Here it is. Hugo Chavez was doing this in Cuba, you may remember, months ago. It didn't seem to help. It wasn't being done for cancer. It was being done for other reasons. But combined with a low glucose diet, or better still, starvation, literally don't eat for a week, plus the hyperbaric oxygen therapy, looks promising. I say looks promising. The clinical data is still very preliminary. We're having a workshop on it in November. I should know more then. But it fits in precisely with what we've been thinking about the oxygen story. The good old diet and exercise, incidentally, is pretty good advice. Get lots of oxygen into your body and watch what you eat. We make another prediction, and so this is just something we thought up on the run a couple of weeks ago, that if cancer, the adaptive immune system dates back, I think, about 400 million years. If cancer, as it progresses, reaches back further than that, it will decouple from the adaptive immune system. So cancer is more vulnerable to infection than healthy cells. So that just suggests that we might vaccinate people against pretty virulent diseases and then infect them and infect the cancer. Kill the cancer and not the patient. That's probably a crazy idea, and I'm just throwing it out there. I haven't discussed it with anyone else, but we're developing these ideas with one of the members of my team who's an immunologist to see if any of this makes sense. The other thing that we can do, I've said we don't have to cure cancer, we just have to manage it. If we can extend the period of dormancy, supposing somebody says to you, I'm dreadfully sorry, you've been diagnosed with cancer. we're going to remove the primary tumour by surgery, but I have to tell you, there's a 90% chance that the cancer will return in 5 years. That's a pretty common story today. The cancer comes back, spreads around the body, and it comes back. Maybe a year later, 5 years, 10 years, sometimes 20 years later. It's lurking there, it's dormant in the body. If we understood what that dormancy process was and could extend it, if somebody said, dreadfully sorry, you've been diagnosed with cancer, we're going to remove the primary tumour by surgery, but I have to tell you there's a 90% chance it will come back within the next 80 years. Worry about something else. So we don't need a cure. We don't even need to understand what's going on inside the cancer. We just need to extend that period of dormancy. Now, I'm coming near the end in case you're getting bored. The extraordinary thing about gene expression is it depends not just on chemical signals, but physical signals as well from the environment. Things like the hardness of surfaces. A cell on a hard surface, like against bone, expresses different genes from a cell in soft tissue. I have a colleague at ASU, she sends cells up into orbit, up to the space station. They express genes differently up there from what they do on the ground. So they're responsive to the mechanical microenvironment. Turns out they're also responsive to electric fields. I'll show you something in a moment. Oxygen tension is another important thing. So all of these things will affect how the genes are expressed. If you take a cancer cell and put it in a normal tissue environment, it will more often than not just behave like a normal cell. Won't make another tumor. Likewise, if you take a healthy cell and put it in a tumor environment, it joins the enemy, defects to the other side, becomes cancerous. So there's a lot about the tissue microenvironment that controls whether cells are healthy or malignant. And we're working with Mina Bissell at the Lawrence Berkeley Laboratory, who is convinced that the cancer is a disease of the tissue microenvironment. If you have a mechanical, electrical, stressed, badly organized tissue microenvironment, that this can trigger cancer. And likewise, if you restore a healthy tissue microenvironment, you can control the cancer. So let me just show you this, because I thought this was really amazing when I saw it. This is the work of Mike Levin at Tufts University, and these things you're looking at here are called planaria. They're little worms that school children like to play with because you can chop them up and they regenerate. And so if you chop them down the middle, the head grows a tail and the tail grows a head. And it turns out they're electrically polarized, plus at one end, minus at the other. And if you muck around with the electric fields, you can make them grow two heads, like this, or two tails. When I first saw this picture, I asked Mike Levin, are these ones with two tails really stupid? And he said, not noticeably so. Now, the point is, so the fancy way of expressing this is that these are different phenotypes. They've all got the same genotype. They all have the identical genes, but they're expressed differently. And the electric fields affect how those genes are expressed. So it's a very good example of top-down causation, because the electric field is something that is spread out over the whole of the organism, but it affects how genes are expressed down at the molecular level. Now, I just got this idea, which is probably half-baked, and I don't suppose there are many people in this audience here who remember Ilya Prigogine, but he was a big name, Nobel Prize-winning chemist, big name in science when I was a student. He developed the theory of far-from-equilibrium thermodynamics. And he coined a term called dissipative structure. A dissipative structure is a system. I'm going to flip over two slides to show you a familiar dissipative structure. It's the great red spot of Jupiter. You don't often see that in a lecture on cancer, but I'm trying to connect all these things. The great red spot of Jupiter isn't a solid object. It's not a fixed thing. There's a throughput of matter and energy. The matter comes swirling in and goes around and goes out again, and it's far from equilibrium. So there's an export of entropy for those physicists in the audience that know what I'm talking about. So this is a system that is in dynamical equilibrium with its environment, far from thermodynamic equilibrium, exporting entropy, throughput, burning energy, throughput of material. So I have a sneaky feeling that there are two classes of cancer dormancy. One is individual cells that are just like spores that maybe sit in the bone marrow, don't do much for a long time. And the other are microtumors, micromets, they're often called in the trade, typically 30 to 50 cells, sitting in a tissue environment, but a little bit like the first colonists of the United States. They all went off in a boat, and they got to the other end, they built a fort, keep the Indians out, and then they sort of suffered a lot of disease, and they were sort of, it was touch and go, and eventually they stabilized, and then they started recruiting the tribes of Indians round about, bribing them with beads and things like that, until they got a sort of critical mass, and then boom, out they burst and took over the whole country, and the rest is history. So I see this as a little bit similar. These micromets are like dissipative structures. They're not inert. They've got a throughput of energy and entropy, and there's probably a turnover of cells. Some are dying on the periphery, knocked off maybe by the immune system or by the tissue microenvironment, but the ones in the middle are protected, and they just keep ticking over. And I think we could build a mathematical model of this. And what we have to understand is, how can we keep them ticking over happily like this for not 5 years, but 50 years? It's all we want to do. Control that physical microenvironment enough to prevent them bursting out of the fort. So this is the only really technical slide. We can imagine many parameters, physical parameters, pressure, oxygen, tension, pH are ones I've mentioned, but there'll be others as well. Maybe half a dozen, pick your favorite ones. And that there'll be some island of stability in there where these micrometastases can sit comfortably. But if something perturbs them, if there's a stress and they perturbed outside of that comfort zone, then they get into this dangerous region, and then they can explode and become full-blown metastatic cancer. Another way of looking at this, and we're coming very close to the end now, this is the phenotypic landscape, but we can imagine there's healthy cells sitting in, so physicists love these landscape analogies, that little ball sitting in a well. So healthy cell, everything's stable down here. Cancer is another stable condition, it's down here, because for the cancer, everything is working just fine. These quiescent dormant cells, I think, or dormant micromets, are sitting in, hung up in some little valley in between. And what we need to do is learn how to manage that landscape in some manner. So I'm just going to give you the conclusions now. Cancer is, people often say it's a modern disease. We must be doing something wrong. It's not a modern disease. It's very ancient. It's got very deep evolutionary roots. It's very widespread. It's part of life, part of the fundamental logic of multicellularity. There are many ways of triggering cancer. We should obviously want to prevent those, but we shouldn't think that the answer to cancer lies into figuring out, well, what caused that? Because lots of things will cause it. Many random ways, but once it gets going, it's very deterministic. So it's that program, that deterministic program we should concentrate on, the one that runs the cancer, probably with all its genes intact, not defective, triggered inappropriately, but running just fine. I think it's mostly a response to a stress microenvironment. That is, I'm a great believer in the epigenetic top-down explanation for cancer. Maybe not always, but I think that's a large part of it. And most of the genetic damage that you see is that instability is just the collateral damage. The cell doesn't bother to edit the mistakes, I think, because of the different ways of managing its energy. The important point is that most of what I've been talking about is cancer is a rerun of embryogenesis, but it's of course not as well ordered as embryogenesis, but it's upregulating those genes. So it's almost as if the cells think, oh, I'm an embryo again, I must replicate, move around, because in the embryo, cells are moving all over the place. It's a very motile environment. Or alternatively, they're thinking, oh, it's just like 1.6 billion years ago, let's have fun. So and that as cancer progresses, cancer stages, not really often studied in cancer biology. The staging of cancer, it starts off, a small tumor and ends up with full-blown metastatic disease. As a function of cancer staging, that we should see a correlation in reverse with these two things. And that we don't need a cure for cancer. We're not going to find one, a general purpose cure. We don't need one. All we need to do is to delay its onset and manage its progress when we've got it. And it's often said that the reason there's so much cancer is people are living so much longer. That's certainly, there's some truth in that. because cancer risk increases with age, and we've cracked a lot of the other problems in the developed world. So people are living longer, and so more and more people, 1 in 3 men will get cancer, 1 in 2 men, 1 in 3 women on current figures. But as people live longer, those figures will go up. But we don't need to cure it, we just need to... There'll be something else waiting in the wings that will be the number 1 killer that we'll all worry about. It could be diabetes, don't know. But you see, we don't have to come up with the pill that will make it go away. So that's the bottom line. I normally give lectures on black holes and the Big Bang and quantum physics and search for aliens and so on. And it's always a lot of fun, and I always end on an upbeat note. It's hard to have too much fun with cancer. I do just want to say that although for me it's a scientific mystery, and I love trying to solve mysteries, I said it before, cancer touches every family on the planet. It certainly touched my family. I think we all are very much aware of cancer. We want to do something about it, and I'm very serious in this quest. So this is not just a bunch of ignorant physicists just entertaining themselves at public expense. I really do take seriously the need for radical new thinking and for controlling cancer in imaginative new ways. And so that's the spirit in which I've been delivering this. If you want to know more, please visit our website. My wife created it, so she's always pleased when there are hits. And there's a lot of information there, not only what I've been talking about, but all of the other more sort of down-to-earth stuff that our centre and the others do. So, ladies and gentlemen, thank you for your attention.
Thank you very much, Paul, for a very thought-provoking idea. We have some time for a few questions, and we've got a couple of roving mics, one down here, one upstairs. So if you hold your hand up, I'll try and get around as many as possible. Why don't we start with the person nearest to you, Lucy?
That was a very interesting talk. Thank you very much, Paul. Just a quick question. The cancer research charities, what sort of form does their research do into the work you're doing? How does it link up?
Right. The second time today I've been asked this. What about the non-profit sector or the non-government sector? All our money comes from the National Cancer Institute. So and this is the first time I've ever received money to work in anything of clinical relevance. And so I really can't tell you, except there are, of course, a large number of foundations. I'm thinking like the Howard Hughes Foundation that will support cancer research. I get the impression that really there is a reigning paradigm. It's dominated by sequencing. Sequencing is guaranteed to give you results because if you buy a sequencer, you can sequence genes till the cows come home. So everyone wants to do that. It's the relevance of it I'm less convinced about. And so there's something called the Cancer Genome Atlas at the moment, which Anna Barker, who I mentioned earlier, is now at ASU, was responsible for getting that going. Terabytes of data pouring out of that. Keeps a lot of people busy, but a very dubious relevance, in my view, to being able to crack the cancer problem. So I'm not sure with the cancer charities whether any of them think that they really need to be putting money into something radically new. I'm not picking up that sense, but maybe some of the smaller ones are prepared to take a gamble on something like this, which is very much outside the box as far as mainstream cancer research.
Are you talking to the cancer research charities?
Well, I'm talking to Cancer Research UK at a retreat. Probably we're forbidden to talk outside the retreat. The ideas are so crazy, I don't know. In July.
Lucy, gentleman down here. Is there anybody up there? Yeah. Kim, would you? Yeah. Oh, yeah, that's fine. Yeah. Let's take this one first.
Thank you very much.
Can you say who you are as well, please?
My name is Jones, Dr. Robert Jones, and I've been in cancer research for 53 years. Oh dear, what was your last point? Yes. Why don't we have a simple treatment for cancer, which we do need? It's because it's been suppressed. In 1974, I had a bright idea that if we could disrupt energy metabolism in the cancer cell, we might be able to control growth. But I was wrong. You disrupt it, you cause necrosis. For a period of time, I've been hammering on the doors of the cancer research charities trying to get support, and I couldn't get it. But in 1996, it was cracked. This is my book, which I sent to the New Scientist for review in 2007. They didn't bother to answer my letter. They sold my book on. Now, how do we do it? When we spoke about mitochondria, Professor Davies, there are mitochondria in the cancer cell, and they are important, and they do function. And if you disrupt oxidative phosphorylation, you can cause necrosis. And you can do it with a variety of drugs. You can do it even with hormones like vasopressin. But the trouble is that you tend to get diffuse necrosis. That's another story. Can I please finish by saying there is a website out there, Energy Grid, 2 words. Sorry, 1 word, Energy Grid, not energy and grid, and cancer. And anybody can have a look at it. And if you're interested in my book, let's talk later.
Thank you very much for that. Just a general comment that I'm persuaded that the metabolic story of cancer and healthy cells is a really important part of what's going on here. And I carefully read, and I know Tom Seyfried, Cancer is a Metabolic Disease, is the title of his book. And some of the things that you've just been saying, I think you will find have been recapitulated in that book as well. So I think there is I think metabolism is a really important part of this story. That's just the general point I want to make. So thank you.
Kim, do you want to pass the microphone? We're up here, Paul, on the balcony.
I'm glad, just as a physicist, again, get involved in current biological processes. I know recently physicists in Cambridge are trying to sequence just by taking the polarity of the genome and then no reagents or anything, and it's a huge step forward. It's a lot cheaper. It's far more efficient. What methods or process could be used from a physicist's point of view then for cancer treatments then?
I'm not sure I caught the first part of that question, but you were saying that what from the physicist's point of view, what better process can be used than
within cancer treatment. So it was just the example that in sequencing, you just get a strand of DNA. You run it through a loop.
Oh, the nanopore, yes.
Exactly, yes.
So the pioneer of nanopore technology is one of the people in my centre, Stuart Lindsay. He was the physical chemist, and he's licensed this to IBM and Roche, and he's developing now single molecule proteomics as well. So this, for the benefit of the people here who don't know about this technology, rather than doing the messy original method of sequencing genomes, if you just take a DNA strand, suck it through a little hole, put some electrodes across the hole, then it turns out, for reasons of quantum physics, you have what's called physicists call a tunneling current across the molecule, and that varies according to whether it's an A, C, G, or T passing through, and you can just sort of suck it through like spaghetti, and then just read off the different tunneling signals that you get. So it's a very good way, in principle, of doing single molecule sequencing. Could work for proteins as well as for DNA. So yeah, that's one way in which physicists are helping by developing technology for diagnosis. What interests me more is where physicists come to the rescue by lending concepts from their subjects, because that's going to be the game changer. Changing the conceptual landscape on which we're approaching the problem. Of course, it's also important that we can help out with the diagnosis and the treatment, but it's only that really major shift in the way we think about cancer. So the great advantage is that if scientists from outside the discipline come in, they can connect up the dots in a different pattern from the people working in the field. And that's what I consider my principal job to be, to survey the whole of the scientific landscape, collect significant facts wherever I see them, look at the cancer field and connect up the dots of the things we know about cancer to tell a different narrative from the one that's being told. So it's the concepts that come from physics are more important than the technology, in my view. But you would expect to hear that from a theoretical physicist, wouldn't you?
Anyone? Lady down here in the third row.
Hello, I'm Philippa Stone, and I had surgery for cancer 5 years ago. It's just a comment and then a question. I'm so relieved to hear you say that stress is, can bring on cancer, because I'm convinced that it brought on mine. And I had an injury as well. I fell, and the tumour grew along the bone, which was fractured in the fall. But my question is about the breast cancer is supposed to be very serious in premenopausal women, but not so much in postmenopausal. But most breast cancer occurs in postmenopausal women. And yet these poor women are put through hell. And, quite frankly, I knew my cancer wasn't life-threatening, but, all my doctors were terrified. I was operated on very, very suddenly.
Right, that's the standard of care.
But I'm just wondering if there's a link between the, the fact that, as you were saying, cancer is linked to the embryonic growth. And, is that the link why postmenopausal women, it doesn't really, speaking for myself, and,
right.
Obviously I don't want to diss anybody who does die from it, but premenopausal, it's very, very serious.
Well, there are a lot of topics in there, and I'm not sure I can address them all or competent to address them all. One thing I will say right at the outset is that I was using the word stress in a technical, scientific sense. So a stressed cell might be one that has the wrong pH in its environment. So a person who is stressed might well, through the circulation of hormones or something, create a stressed tissue environment. But the two don't necessarily go together. It might well be that a stressed person actually is releasing chemical signals that are actually improving their situation. So there's not a one-to-one correlation between a person being stressed and a cell being stressed. That's the first point. The second point is that there is a class of breast cancer that does afflict younger women, as we all know. When I was at the University of Newcastle, there was a young girl of 16 came to see me, Emma Bakes, and she wanted to study physics, and she did, and she came to Newcastle, and she went on and became a physicist and an astrophysicist, and she did that for a few years, and then she decided she wanted to train to be a medical doctor, so she switched careers, and she was in her 5th year, about to qualify as a physician at Stanford University, a little boy of 2, and she emailed me, knowing I was going to be in the area, said, hope we can get together, it might be the last time you see me, I've been diagnosed with breast cancer, I've probably only got a few months to live, and sure enough she died a few months later, leaving this little boy. So 42. Total tragedy. So if ever I think, this cancer research is a lot of fun, I have a picture in my office and see it every day. So it's not just a disease of aging. There are different types of breast cancer. The one general point I can make is that cancer equals inflammation. With the menstrual cycles, and in particular with childbirth and milk production and then weaning, these are big structural changes for the internal ducts of the breast. And all of those changes carry a risk. And so it's, in terms of protection against breast cancer, it's good to have lots of children and breastfeed them for a long time. As to why it's postmenopausal women, my guess, but this is just standing up here off the top of my head, I would guess that the damage is done at an earlier stage, but the regulatory mechanisms that are in place in normal tissues continue to hold it in check. And so it's latency. It's a period of latency. That word did appear on one of my slides. So there's latency and there's dormancy. Latency is the period of time you wait before the cancer first embarks on its great journey. Dormancy is what happens if you intervene and then it recurs. And so I think it's that long period of latency, and we don't understand that any more than the period of dormancy, but it will be something to do with the tissue microenvironment And I do know very well the people working on this particular problem.
Thank you. We've got time for a couple more questions. So we'll do one up here and then we'll come down, Lucy, behind you.
Thank you. My name's Martin Williams. And the mic that it's working now, thank you. Right, 2 questions. Briefly, we've already touched on mitochondria. To what extent are mutations in mitochondria responsible for development and propagation of cancers? The second question is, you've already used the word subroutine many times, and you've already mentioned extensive DNA sequencing. To what extent are analogues in the digital world, in other words, in the discipline of computer science, useful in understanding cancers?
Well, I'll answer the second one first. I was much struck by an article in Nature by Paul Nurse, who's currently the president of the Royal Society, on life, logic, and information flow. And I think the analogy with modules in an electronic circuit is a very good one in biology, in cancer biology as well. And that's one way that we will understand things like the transition from single to multicellular life, the transition to cancer and so on, is the change in the management of the information flow. And in fact, I just was awarded nearly $1 million for a grant for young Sarah, I showed her picture there, to work on the origin of life as a transition in the information management of the system. So I think there's really deep links in the flow of logic. Another thing I can mention is one of the people in my centre, who's now moved to Dundee, has found that if you look at the topology of the gene regulatory network, because genes switch each other on and off and they form a complex web. If you look at the topology of that web, for reasons of stability, it's got to have certain topological features. In the data he's been able to get so far, it looks like it's different in cancer from in healthy cells. So we're really here talking a bit like the internet or something. So it's again a sort of computational analogy. I think it's really very useful. And I've answered that at such length I've forgotten your first question.
First question was, you've already touched on mitochondria, the gentleman.
Oh, the
below.
mitochondria, yeah.
But to what extent is
right.
mutations responsible?
Yes. So I'm not trying to say the mutations are never part of the story in cancer. I think there are 2 types of mutations. One is those that will trigger the cancer, and there could be a wide range of those. And then there are those that are just the collateral damage. And I can well imagine that a mutation in a crucial mitochondrial gene causes a dysfunction of the mitochondria, which then brings on, via this whole sort of Warburg effect, a change in the metabolic regime of the cell. Now, of course, one mitochondrion, most cells have many, not sure the knock-on effect, but I think this is I think the story of oxygen oxidative stress, and I should mention, incidentally, because just tomorrow, Jim Watson, he of the DNA, double helix, will be talking at the Cheltenham Literary Festival at the exact same time as me, so I won't have any audience at all, I'm sure, for my exoplanets. And he is going on and on at the moment about how cancer cells don't handle oxygen well because of their different metabolism, and that we need antioxidants for cancer patients, because that means that the cancer will die of the oxidative damage faster than the healthy cells. So I don't know the answer, but I think it's a really important line of inquiry. I mentioned our next workshop is on exactly this stuff. It's on the Warburg effect, cancer metabolism, on oxygen, with the same Tom Seyfried, whose name I just mentioned. So more to follow. Well, I'm not sure about start smoking, but go and take hyperbaric oxygen therapy, I would think.
Okay, Lucy, lady down here.
Yeah, I think we need to take this.
If you can hang on and talk to Paul later. Yeah.
Hi, thank you for your talk. My name's Marianne Baker. I have a PhD in tumour microenvironment and angiogenesis. I thought you put a lot into a talk, and while you said there were no black and white issues in biology, which I completely agree with, a lot of them were presented as such, and I think there was a bit of possibly dangerous information in there. And I do have a question at the end,
right.
So things to kind of get
Having
there.
been suitably wrapped over the knuckles by a professional biologist, which I am absolutely not.
So I think you conflated what works with what's being designed, or you said determinist kind of view, cancers are actually incredibly bad at what they do. You're completely right, they are monster cells, and while they are deadly and they're good at spreading and doing that, they do kill you in the end, and therefore they die. So they're not perfect, and they're certainly not deterministic, I wouldn't say at all. So mutations lift the blocks that are there to stop cells proliferating uncontrollably.
Right.
And it's not necessary, as you said, there are passenger and driver mutations, as they're called in cancer biology. So the driver mutations occur and allow cancer cells to proliferate, and as they do so, they acquire more mutations. So the more they proliferate and the more mutations they get, the more mutations that occur will be driver mutations. So while there can be a lot of passenger mutations that don't do anything, it's like evolution, and the cancer cells that get better at proliferating are selected for.
Right, you're articulating very well the standard paradigm, which I don't believe is more than a perturbation on the main story.
Offering the other view, which I think personally, and most of cancer biologists think makes more sense, and I know it's your job to provide a different view, but just to explain that. So cancer, solid tumours in particularly, create hypoxia, the lack of oxygen. It's not that they hate oxygen, it's that when you have a big mass of cells, like a bunch of people in a room, they don't have enough. And that's what brings blood vessels in. That's what brings blood vessels in, and that can help to fix hypoxia, but it doesn't do it very well, again. And since the 70s, there has been an idea that if we can make tumour blood vessels better and bring more oxygen in, then chemotherapy will do better as well, because it can get to the tumour cells.
Yes, I'm familiar with that.
So
you've got a long list there.
Yes, sorry. So, but that's specific to solid tumours. What is your point of view if you think oxygen is the way to fix cancer, which I completely disagree with? What's your view in hematological malignancy? So the cells that make up the blood, which then flows around the blood, which is full of oxygen when it's red. Hematological malignancies like leukemia, lymphoma, how would you propose treating them? Why do they succeed at all?
Yes, I don't have an answer to the hematological puzzle, and I'm not saying that there's a one-size-fits-all story here. I'm convinced that oxidative stress is a really important part of this story, and the cancer is more vulnerable, and I share that opinion with Jim Watson, so I suppose I'm not in terrible company. Well, some people don't like Jim Watson. A lot of people don't like Jim Watson. But I'm pleased that you picked up on this business about, you see, you're following the party line. There are all these wonderful survival traits that cancer has, just happen to be co-located in the same neoplasm, and they arise from Darwinian evolution of the neoplasm in a period of weeks or months. They just happen to stumble across all of these things. And I don't buy it, and I realise that is trotted out as the party line. I'm paid to think in a contrarian manner. I think that these survival traits are pre-programmed and that they are triggered by these changes. So, as you quite rightly pointed out, mutations in tumour suppressor genes can lift the lid off, and the program can then run. I think the majority of the accumulating mutations that arise after that, it really is a mess by the late stage. Almost all of those are irrelevant, and I think the reason they arise is because cancer is running on this different metabolic cycle, and it's allocating its energy resources differently. It isn't bothering to divert the resources to all the error correction, so that just goes to pot.
It's well known that it's a problem of a lot of cancer cells, and it's a hallmark that error correction is one of the mutations you need to get more hallmarks. So it's a selection process. If you happen to get
yes, I just say nobody's ever done the mathematics convincingly that you will get all of those properties in the Yes, but you'd have to convince me, and you'd have to show me the mathematics, that that is enough to give you, in the same population, all of those different traits. But, you see, we don't need to worry about that, because we're making definite prediction about as cancer progresses, how it will correlate inversely with the phylogenetic tree. If you tell me your prediction, then we have a means of testing these alternative points of view. I've never heard any prediction of that nature coming out of the standard, make it up as you go along, do the evolution on the run. Absolutely, there's a lot of... Right, That's why the whole, in my view, mainstream cancer research is charging down the wrong path. They're focusing on exactly the wrong thing. They're focusing on the differences, on the heterogeneity. They should be focusing on the conserved parts of the genome. That's running the cancer. If you hit that, then you really have got something. Sorry? I think we've
So perhaps I can draw this to an end, because it sounds to me like it sounds to me like this is a discussion for later.
It's an important point.
I can see, and I understand why it's such a controversial theory as well, and difficult for biologists to accept. That's the. I'd like to call it to an end this evening. I'd like to thank everybody for coming this evening. Please do go and take a look at the New Scientist stand outside. Wonderful one-time only offer, as I mentioned earlier. Could we please thank the speaker, Paul Davies.